Good day, class. For today's topic, we will be discussing about bilirubin determination. At the end of this laboratory briefing, the students must be able to know a brief background about bilirubin, where it came from, how is it metabolized in the body, all sorts of things. I know this has been taught to you in the lecture, but we will have a reinforcement about this. We will also discuss the different methods in order for us to determine bilirubin in the laboratory, to determine and measure the direct and total bilirubin, and also how to calculate and determine the indirect bilirubin. I will also be teaching you the different reference range of bilirubin among adults as well as among children. So what is bilirubin metabolism? This is a process by which heme, remember that's a component of hemoglobin, and also other heme-containing proteins is degraded and converted into bilirubin, then conjugated, transported, and excreted primarily via the liver into the bile and ultimately in the intestine. This process class is very crucial for number 1, recycling of heme iron. Number 2, that's detoxification of potentially harmful heme-derived products. And number 3, maintenance of bile pigment homeostasis. The major source of bilirubin in our system class is from the turnover of senescent red blood cells, with approximately around 80 to 85% coming from hemoglobin degradation, while the remainder of that percent comes from the breakdown of other heme-containing proteins such as cytochromes and myoglobin. This process occurs primarily in the reticuloendothelial system, mainly the macrophages of the spleen, the Kupffer cells from the liver, and bone marrow, where heme is enzymatically converted into bilirubin through 2 main steps. And what are these steps? Okay, kindly see this picture. Can you see it clearly? In the first step class, you can see the enzyme heme oxygenase cleaves the heme ring in the presence of oxygen and NADPH. What's NADPH again? That's nicotinamide adenine dinucleotide phosphate hydrogen. And then it produces biliverdin. The free ferrous iron class and the carbon monoxide, you can see there, that's the only endogenous source of carbon monoxide in humans. Okay, think about this. The biliverdin, that's a green pigment, will be reduced to bilirubin, which is the yellow-orange pigment, by the enzyme biliverdin reductase in a reaction that also uses NADPH. Can you see there clearly? Alright. Now, this type of bilirubin class, also known as the unconjugated or the indirect bilirubin, is lipophilic and is insoluble in water, which poses a potential toxicity risk if not properly handled by our bodies. Once produced class, this unconjugated bilirubin will bind tightly but not covalently to plasma albumin for transport through the bloodstream to the liver. So, it means that the unconjugated bilirubin must be bound to plasma albumin. And this binding makes bilirubin more soluble enough to circulate and prevents it from crossing cell membranes. That includes the blood-brain barrier, because we all know that that is crucial because free bilirubin is neurotoxic. Upon reaching the liver class, bilirubin dissociates from, it's bound to albumin, remember? So it dissociates from albumin and it enters hepatocytes through a facilitated diffusion, which is mediated by transport proteins. Inside the hepatocytes, bilirubin then binds to cytosolic proteins such as ligandin, or glutathione S transferase B, which keep it soluble and prevent it from diffusing back into our bloodstream. The next critical step is conjugation. This occurs where? In the smooth endoplasmic reticulum, correct? In here, class, the enzyme uridine 5-diphospho-glucuronylyl transferase catalyzes the covalent attachment of one or two molecules of glucuronic acid, which is derived from the UDP glucuronic acid, to bilirubin. The resulting products, that's bilirubin mono and diglucuronides, are water-soluble and then termed as conjugated or direct bilirubin. This transformation is essential for safe excretion as conjugated bilirubin is no longer lipophilic. Then, after that, we all know already that the conjugated bilirubin is actively secreted from hepatocytes into the bile canaliculi through an ATP-dependent transporter, which is known as the multi-drug resistance associated protein 2, or the MRP2. From there, it travels with bile into the bile ducts and is released into the small intestine. And from there, in the small intestine class, the conjugated bilirubin undergoes further transformations by our bacterial enzymes. So, the beta-glucuronidase produced by our intestinal flora hydrolyzes bilirubin diglucuronide back to unconjugated bilirubin, which is then subsequently reduced by anaerobic bacteria to form urobilinogen. This urobilinogen class is a colorless compound. So as a recap, biliverdin again is the green pigment. For bilirubin, that's the yellow-orange pigment, while the urobilinogen is the colorless compound. This urobilinogen class has several possible fates. The first one, the majority of it is oxidized by intestinal bacteria, and it is formed into stercobilin, which, by the way, actually imparts the characteristic brown color of our feces, remember? While a smaller fraction of urobilinogen is reabsorbed from the intestine back into the portal circulation. Most of this is taken up again by the liver and re-excreted into bile, forming part of the enterohepatic circulation. A small portion of urobilinogen escapes hepatic uptake. It enters back into the systemic circulation and is filtered by the kidneys. And so, in the urine, this urobilinogen class is oxidized to urobilin, which gives our urine its yellow color. So, in summary, the bilirubin metabolism represents an elegant and essential detoxification pathway. Again, heme is degraded into biliverdin and bilirubin. Bilirubin is transported to the liver bound to what protein? That's albumin, conjugated to increase its solubility, and then secreted into bile, and finally transformed in the intestine to urobilinogen and stercobilin for excretion. This continuous cycle not only clears potentially toxic heme products, but it also contributes to the pigmentation of our feces and urine, making bilirubin metabolism both physiologically vital and clinically significant. We have been discussing about the bilirubin metabolism, but what really is bilirubin? Bilirubin class, it is an orange-yellow pigment formed from the breakdown of red blood cells. So, when hemoglobin from senescent red blood cells is degraded, it releases heme, and this heme is converted to biliverdin and then bilirubin. It's also very important clinically because high levels cause jaundice. The first person who discovered this is Rudolf Virchow. And Rudolf Virchow is a German pathologist, often called the father of modern pathology. He discovered this pigment in old blood clots and he calls it hematoidin. At that time, Virchow didn't realize that hematoidin and bile pigments were related. Next is Stadler. Stadler is a German chemist which gave the pigment its current name, bilirubin, for the very reason that when he combined bili, that means bile, and rubin means red, and he identified that the pigment Virchow had found was the same one found in bile. Next is Ivan Tarchanov. He is a Russian physiologist and he demonstrated the direct association of bile pigments like bilirubin with hemoglobin. He showed bilirubin comes from hemoglobin breakdowns linking red blood cell destruction to bile pigment formation. And then we have Hans Fischer, who is a Nobel Prize winner in 1930 for work on heme and chlorophyll. He also has a collaborator, and that is Heinrich Pleininger. Their contribution is that synthesized bilirubin 9A and he proposed its chemical structure. Their work clarified how bilirubin is built from the tetrapyrrole ring structure, which has the same core as a heme. So, to summarize, Virchow viewed a strange pigment and called it hematoidin. Stadler set its name, which is widely known now as bilirubin. Tarchanov tied bilirubin into hemoglobin, while Fischer and Pleininger formed and proposed its structure. Now that we have an idea about what bilirubin is, let's now see the difference between conjugated and unconjugated bilirubin. As discussed earlier, we know that the chemical composition of an unconjugated bilirubin is that it is not soluble in water and it is lipophilic. Compared to that of conjugated bilirubin, it is water-soluble but less lipid-soluble. The conjugated one is transported in blood in a free form, while the unconjugated bilirubin is bound to albumin. The unconjugated bilirubin is still processed in the liver by being conjugated with glucuronic acid, while the conjugated bilirubin is already processed and it's ready for elimination from the body. The unconjugated type of bilirubin can pass through the blood-brain barrier and cause neurotoxicity. Next, since conjugated bilirubin is already ready for elimination from the body, it is excreted in the urine. Compared to that of unconjugated bilirubin that still needs processing, it will not be excreted in the urine. The conjugated form class does not contribute to the yellowish coloration of the skin or even the eyes, so it's not responsible for jaundice compared to that of unconjugated bilirubin, which is responsible for the yellow color of bruises as well as jaundice. And you can see here the difference between conjugated and unconjugated bilirubin, that unconjugated bilirubin is typically elevated in conditions such as hemolytic anemia and Gilbert's syndrome. For conjugated bilirubin, it's typically elevated in conditions such as obstructive jaundice and biliary atresia. Also, please familiarize yourselves with the other names of indirect or unconjugated bilirubin compared to that of direct or conjugated bilirubin. But actually, class, some of the terms are obvious, quite obvious, because you can see that unconjugated or indirect bilirubin is also known as water-insoluble bilirubin, right? So it was already discussed earlier. Can also be called non-polar bilirubin. It can also be called hemobilirubin because, as we all know, that unconjugated bilirubin is derived from heme. Remember that heme, it's the iron-containing part of hemoglobin. Why do you think it's also called slow-reacting bilirubin? Aside from it is not water-soluble, it does not react unless alcohol or any other solvent is used in the reaction. And lastly, it can also be known as pre-hepatic bilirubin because it is processed outside of the liver. So there are actually reasons why their names are like this. And very lastly, I forgot to mention that indirect bilirubin or unconjugated bilirubin is also called B1, or the bilirubin that is the first that is produced after hemoglobin degradation. Let's now proceed to B2, or the direct or unconjugated bilirubin. Discussed earlier, it says there that it is water soluble. So it's the same. It can also be called polar bilirubin. So why do you think it's also called cholebilirubin? Let's try to break it down, class, so that you can understand. The prefix chole, it comes from the Greek word chole, meaning bile. And then its second name is bilirubin, which is the yellow pigment produced from the breakdown of heme. So, cholebilirubin literally means bile bilirubin. B2 is also known as 1 minute or the prompt bilirubin because it reacts quickly because what's the rationale behind it? It's water soluble. And so, it can react directly with the reagent during the reaction. And lastly, it's also known as a post-hepatic or hepatic or obstructive and regurgitative bilirubin because it is found during, or it is created in the liver, it's conjugated in the liver, and can also be found post-liver or post-hepatic phase. Another form of bilirubin would be delta bilirubin. It is also called biliprotein, and it is a form of conjugated bilirubin that is covalently bound to albumin in the blood. Normally, class, we know that conjugated bilirubin or the direct bilirubin is water soluble and it is excreted into the bile. However, when bile flow is obstructed, examples would be if there is cholestasis or if the patient has hepatitis, this conjugated bilirubin leaks back into the bloodstream. And once in the blood, some of this conjugated bilirubin forms a stable, irreversible covalent bond with albumin. This complex is then called the delta bilirubin. So if you ask me, Miss, is this delta bilirubin water soluble? The answer would be yes, because it is conjugated. And then, this delta bilirubin class is measured as part of direct or conjugated bilirubin in laboratory tests. Okay, so in order for us to identify or to measure the total bilirubin, we would have to add both the conjugated bilirubin and the unconjugated bilirubin, and you add also the delta bilirubin. But in the laboratory tests class, the delta bilirubin will automatically be counted as part of the conjugated bilirubin. So there's actually no need for us to separately compute for the delta bilirubin. What is detected in the laboratory machines would be the conjugated bilirubin and the total bilirubin. And in order for us to get the unconjugated bilirubin, we have to subtract total bilirubin and conjugated bilirubin to get the values for the unconjugated bilirubin. Is that clear? Let's now proceed to the different diseases associated with elevated levels of total bilirubin and indirect bilirubin. Let's understand further the main mechanisms as to why these diseases would happen and which mechanism do they belong. Number one would be increased bilirubin production. This means that there is too much bilirubin from excessive RBC breakdown or from hemolysis. Can you help name the example diseases for this mechanism? That would be hemolytic anemias, either autoimmune, due to hereditary spherocytosis, can also be from sickle cell anemia or from thalassemia. It could also be due to transfusion reactions or hemolytic disease of the newborn. Another mechanism would be impaired hepatic uptake. This means that the liver cells fail to take up bilirubin from the blood. Examples would include sepsis, or if the patient has a congestive heart failure, meaning there's reduced hepatic perfusion, can also be due to the intake of certain drugs like rifampicin or probenecid. And the very last mechanism could be due to defective conjugation. This means that the liver can't conjugate bilirubin properly. The first example disease would be Gilbert syndrome. This one is a common, mild, inherited liver condition that causes slightly elevated levels of unconjugated or indirect bilirubin in the blood. This is a benign genetic disorder caused by reduced activity of the enzyme uridine 5-diphosphoglucuronosyl transferase, or the UGTA, which is responsible for conjugating bilirubin in the liver. Since this enzyme works less efficiently, the unconjugated bilirubin then accumulates in the blood. Next would be the Crigler-Najjar syndrome type 1 and type 2. This is a rare inherited disorder of bilirubin metabolism caused by a defect in the enzyme UDP glucuronosyl transferase, or the UGTA. That's the same enzyme that's partially reduced in the Gilbert syndrome, if you can remember, right? But in Crigler-Najjar syndrome class, it is severely deficient or absent. So, the conjugation step fails in this syndrome, thus, the unconjugated bilirubin accumulates in the blood. Which among the two types of Crigler-Najjar is severe? That is type 1. Because in type 1 Crigler-Najjar syndrome class, the enzyme is totally absent or 0%. But for type 2, it's milder. It's reduced around less than 10%. So for those that have my notes, these are the corresponding diseases that would happen if there are elevated levels of total bilirubin and also elevated levels of indirect bilirubin. Kindly familiarize yourselves with the difference. It's rather obvious that if the reason is prehepatic or hemolytic, indirect bilirubin would be elevated. Examples would be hemolytic disorders, sickle cell anemia, thalassemia, or the liver diseases resulting in impaired transport or conjugation within the liver. If the problem is hepatic, that means conjugation defect, then that would be Gilbert syndrome, Crigler-Najjar, or neonatal jaundice. If it is drug-induced, then it is due to the intake of rifampicin and probenecid. Let's proceed to the analysis of bilirubin. The goal of bilirubin analysis is to measure total, direct, and indirect bilirubin in biological samples, could be serum or plasma, and also urine, using chemical color reactions. As you already know, class, the earliest and most widely used principle is the diazo reaction. This classic diazo reaction by Ehrlich in the year 1883 uses the principle when bilirubin reacts with diazotized sulfonilic acid, which is, by the way, made by reacting sulfonilic acid plus sodium nitrite plus acid to form a red or purple colored compound called azobilirubin. The intensity of the color is proportional to the bilirubin concentration. During this time class, Ehrlich used urine samples. And this reaction could only detect conjugated bilirubin because unconjugated bilirubin is not water soluble and doesn't react unless an accelerator is added. So the limitations for this is that it is not suitable for serum and it couldn't measure indirect bilirubin because there's incomplete picture of bilirubin metabolism. Next would be the discovery by Vandenberg and Mueller, which adapted the diazo reaction to serum samples and discovered two patterns of color formation. The first type would be the direct reaction or the prompt. The reaction would take place within 1 minute, so there is immediate color formation, which means that this indicates that the conjugated bilirubin is detected because it is water soluble. The next type of reaction would be the indirect reaction, or otherwise known as the delayed reaction. This means that no immediate color is presented or formed, but it reacts only after adding alcohol or the accelerator. This indicates that the unconjugated bilirubin is now detected, which beforehand is insoluble without the accelerator. The third one would be the Malloy and Evelyn method during the year 1937. This now serves as an improvement from the first two diazo reactions, and they created the first clinically useful quantitative test for serum bilirubin. Its principle is the same as that of the Ehrlich's diazo reaction, but Evelyn and Malloy used 50% methanol as an accelerator or solubilizer to allow both conjugated and unconjugated bilirubin to react. The color intensity or the azobilirubin form was then measured using a colorimeter or a spectrophotometer at around 540 to 560 nanometers. The procedure follows: you obtain the serum sample first and then add the diazo reagent. When you add this reagent, it forms a color reaction with conjugated bilirubin. So now you got the direct bilirubin. And then after that, you add the 50% methanol. This step is very crucial because it solubilizes the unconjugated bilirubin. Therefore, you now have the value for total bilirubin. So in order for us to calculate the indirect bilirubin, just simply subtract total bilirubin and direct bilirubin. Advantages of Malloy and Evelyn procedure includes reliability and practicality for routine clinical use, and it's used in laboratories for decades. Lastly would be the Jendrassik-Grof method. The principle with this would be still the same for the diazo reaction, but it is more refined and more accurate version. This is considered the candidate reference method published by the American Association for Clinical Chemistry and the National Bureau of Standards. The modified Jendrassik-Grof method is more complex compared to that of the Malloy-Evelyn procedure, but this method offers more advantages because it is not affected by pH changes. So it's less sensitive to pH in light. It can produce a stable color because it is less fading. So it maintains optical sensitivity even at low bilirubin concentrations. With that, it means that it is suitable for automated analyzers. It has minimal turbidity and relatively constant serum blank. It's also insensitive to a 50-fold variation in protein concentration of the samples, and it is not affected by hemoglobin even up to 750 milligrams per deciliter. Also, the reagents for modified Jendrassik-Grof method class is different because it makes use of caffeine benzoate acetate mixture as its accelerator or solubilizer. The caffeine binds bilirubin, making it soluble in aqueous solution, while the benzoate and acetate buffer maintain stability and the pH. After the reaction, ascorbic acid stops or quenches it, and the alkaline tartrate shifts the color to blue, which makes it stable for spectrophotometry. As for its procedure, the conjugated bilirubin reacts directly with the diazo reagent. You add the accelerator, which is the caffeine benzoate acetate. So this means that it can now detect total bilirubin. You may now then measure absorbance spectrophotometrically. For the specimen collection and storage, this is, by the way, true to all the procedures discussed earlier. Makes use of serum or EDTA or heparinized plasma. But when asked if what would be the answer for our practical examination, it should be non-hemolyzed, protected from light, fasting serum sample. This one is preferred for Malloy-Evelyn procedure. You also have to take note, class, that for Malloy-Evelyn procedure, the addition of alcohol in the analysis can precipitate proteins and may cause interference with the method. Why should it be specific for fasting serum sample? Because the presence of lipemia would increase bilirubin measurements. And it is also indicated that it should not be hemolyzed because hemolysis will decrease bilirubin concentration. And the protected from light, it is because bilirubin will be reduced by 30 to 50% per hour. But if the analysis is not available ASAP, serum or plasma should be separated from the cells and must be stored in the dark. This can be stable for 2 days at room temperature, 1 week at refrigerator temp, and indefinitely at negative 20 degrees Celsius. This now means that bilirubin analysis helps diagnose the type of jaundice your patient may have. When there is prehepatic problems, that would mean indirect bilirubin would elevate or increase. If the problem is hepatic, then both indirect and direct bilirubin is increased. But if the problem is post-hepatic or there is obstructive jaundice, then that means that direct bilirubin is increased. But for the laboratory activity that we will be doing now, we will be using the manufacturer's insert, and it makes use of the modified method of Pearlman and Lee. This is actually an improved adaptation of the classic diazo reaction used for bilirubin determination. The principle that they use is that bilirubin glucuronide, if you can remember, that means the conjugated or direct bilirubin, right? This reacts directly with sulfodiazonium salt, which is formed in situ with sulfonilic acid and sodium nitrite to produce a colored compound, azobilirubin. The color intensity of azobilirubin is measured spectrophotometrically and is proportional to the bilirubin concentration. For the modified method of Pearlman and Lee class, it uses a surfactant as a solubilizer or accelerator, replacing older solvents like methanol or the caffeine benzoate. It helps unconjugated bilirubin, which is water insoluble, to dissolve and react, allowing measurement of total bilirubin in the same assay. The common surfactants that are used could be SDS or sodium dodecyl sulfate or similar detergents. But essentially, the surfactant allows both direct and indirect bilirubin to react smoothly without separate accelerators. The colored product, or the azobilirubin, is measured spectrophotometrically at a wavelength of 540 to 550 nanometers, which, by the way, usually used in the laboratory is 546 nanometers. The features and advantages of Pearlman and Lee method class is that it is already modernized, which uses surfactants. It's safer and more stable than methanol or the caffeine reagents. It also produces a stable color reaction, which is suitable for automated analyzers. It can measure both direct and total bilirubin in one procedure, and it's environmentally safer because it avoids organic solvents like methanol. In order for us to determine total bilirubin using the modified method of Pearlman and Lee, I will already be preparing the working reagent for you, so you only have to pipette 2 ml or 2000 microliters of the reagent and 100 microliter of the sample or 0.1 ml of the sample. You have to mix and incubate for 5 minutes at 37 degrees Celsius. Then measure the absorbance of the sample and the standard against the reagent blank. The factor that we will be using for total bilirubin determination is 24. While if we're already going to detect direct bilirubin, still the same, you have to pipette 2000 microliters or 2 ml of the working reagent and pipette 100 microliter or 0.1 ml of the sample. Mix and incubate for 5 minutes at 37 degrees Celsius. Measure the absorbance of the sample and the standard against the reagent blank. The factor to be used is 17 for direct bilirubin. I have already prepared the working reagent for you, class. All you have to do is to store and handle this with care and at the same time pipette the right volume. This working reagent is stable for 7 days at refrigerator temp, that's 2 to 8 degrees Celsius, and make sure that it is protected from contamination and direct light. Our samples are stable for 1 day at room temperature, that's 15 to 25 degrees Celsius. Can be stable for up to 7 days at refrigerator temp, that's 1 to 8 degrees Celsius. It is stable up to 3 months if we freeze it at negative 20 degrees Celsius. And make sure, class, that you only freeze your samples once. Freeze thawing will destroy our analytes. But remember, class, that one characteristic of a bilirubin is that it is easily degraded. So you must run and analyze the samples ASAP. As for our assay procedure, class, the wavelength to be set in our spectrophotometers would be at 546 nanometers. The working reagent to be pipetted will be 2000 microliters or 2 ml. And then the sample would be 100 microliter. Mix and incubate for 5 minutes and place it in a water bath that is already preset at 37 degrees Celsius. What's the rationale again why we preset our water baths at 37 degrees Celsius? It is to mimic the normal human body temperatures. You're already very proficient with this. You already know how to calculate for the absorbance of total bilirubin and direct bilirubin. Same as how you detect for the other analytes, right? Okay, so it is just change in absorbance of the sample over change in absorbance of the standard. You multiply it with the concentration of the standard. But since our manufacturer provided a factor instead of the concentration of the standard, the way to compute for the value for total bilirubin and direct bilirubin would be this way. Can you remember earlier that for total bilirubin, there was a given factor and that was 24. And also for direct bilirubin, the given factor was 17. So that is how we will be computing for the bilirubin values for today. Just simply substitute the absorbance of your sample and then substitute the number that was given for the factor. And that is already your total bilirubin and or your direct bilirubin values. This is how we calculate for indirect bilirubin. It's very easy, just have to subtract direct bilirubin from total bilirubin. For the instrumentations that we use in the laboratory class, it is already pre-computed for us, so there is no need for us to manually calculate for this. So let's say, for example, the total bilirubin value of your patient is 1 milligram per deciliter, and then the detected direct bilirubin would be 0.2 milligrams per deciliter. So the computed or calculated indirect bilirubin would be 0.8 milligrams per deciliter. Bilirubin estimation usually for total and direct bilirubin is very sensitive to interference from substances in the sample. That's why our first source of error here would be hemolysis and lipemia. It should always be, as much as possible, be avoided as they will alter bilirubin concentrations. Can you tell me why? So when we talk about hemolysis, it's a breakdown of red blood cells, right? And when red blood cells rupture, they release hemoglobin and other intracellular components into the serum. Am I correct? Right. So what happens there is that the free hemoglobin class will absorb light in similar wavelength which is used for our bilirubin assay. So this can falsely increase or decrease bilirubin readings depending on which assay method is used. The heme portion also class of the hemoglobin that is released can oxidize bilirubin to biliverdin, causing falsely, very good, it's falsely low bilirubin values. So let's say, for example, if you use enzymatic methods in order for you to detect bilirubin, then the diazo or the colorimetric methods can also be inhibited by substances released from our lyse cells. How about lipemia? What does lipemic samples do? Remember that lipemia gives the serum a milky or turbid appearance due to high concentrations of triglyceride-rich lipoproteins. And in doing so, turbidity scatters light, which affects absorbance readings in colorimetric or spectrophotometric assays. This leads to falsely high, can also be falsely low bilirubin values depending on the instrument and the wavelength used. But in direct spectrophotometric methods class, lipemia increases the background absorbance, making accurate determination impossible. So if there's optical interference, then there will be inaccurate bilirubin concentration. Next would be, we have to protect samples from light immediately after collection. In the laboratory, we still do this. We wrap our red top tubes or our samples in aluminum foil or use amber colored tubes. And then we store it in the dark or in covered racks until analysis. But as what I have said earlier, we have to analyze promptly as bilirubin should ideally be measured within a few hours after collection. Also, avoid repeated light exposure during handling or even during centrifugation. After processing your samples, these are the expected values using the reagents that we have in the laboratory. If you notice, class, the expected values from our reagents compared to that from our reference textbooks, there's only just a slight difference between the values. So I just want everyone to familiarize themselves with the reference range, either from the expected values of our reagent or from the reference range provided by the textbook. This is another reference range for bilirubin in both adults and infants. You can see this in our reference textbooks from Bishop. My reference for this is Bishop. Again, as what you have seen here, in adults, the conjugated bilirubin is around 0 to 0.2 milligrams per deciliter, while the unconjugated bilirubin must be around 0.2 to 0.8 milligrams per deciliter, and the expected total bilirubin among adults would be 0.2 to 1 milligrams per deciliter. Why do you think, again, premature infants and even full-term infants have a higher reference range for bilirubin? It can be due to their immature livers, or their livers are not fully developed yet. Can also be excessive breakdown of red blood cells from their system, or there could be problems with the elimination as well as in the processing of bilirubin from their immature bodies. So please don't forget all of these facts regarding why kids or infants in general have a higher bilirubin reference range compared to that of the adults. Thank you so much, class, for listening. I hope you've learned a lot from me today. Please keep safe and take care of yourselves. Bye-bye.